Hello and welcome to Dr. Anima Upadhyay's Chemistry Lab videos. In this video, we will try to discuss possible viva questions based on pHmetery. And your exercise for this video is determination of pKa of a given weak acid using pHmeter. So when you are doing this experiment, the first question that could be asked to you is, what is a weak acid? And your answer will be, a weak acid is one which is partially dissociating. What are the examples of weak acid? Give two examples of weak acid. Acetic acid and formic acid are two examples of weak acid. Can you name some strong acids also? Yes, hydrochloric acid, sulfuric acid, nitric acid. These are the examples of strong acid which are commonly used in laboratory. What is pH? pH is negative logarithm of hydrogen ion concentration. What is pK? pK is negative log of acid dissociation constant. How pH and pK are related? pH and pK are related to each other with the help of Henderson-Hasselbalch equation, which says that pH is equal to pKa plus log to the base 10 salt by acid. And with the help of this equation, we can relate pH and pK. So at half equivalence point, pH is equal to pK. Because at half equivalence point, salt is equal to acid. What if the pK of a given acid is very low? What does it suggest? If the pK of a given acid is very low, it suggests that the acid is strong, because pKa suggests the strength of the acid. So what is the difference between pH and pK? pH is the hydrogen ion concentration of a system, and pKa is the dissociation constant. That is, it suggests the strength of the acid. This is the reason a high pH means alkalinity, a low pH means acidity. Whereas a low pKa means a very strong acid and a high pKa suggests that the acid is weak. It shows the strength of the acid. If you are doing pHmetery, what electrodes you are going to use? The electrodes employed to do pHmetery are a reference electrode, which is usually a saturated calomel electrode, and an indicator electrode, that is the glass electrode. So you are saying you are using a reference and an indicator electrode, and you are using glass electrode as the indicator electrode. Right, I am using glass electrode as the indicator electrode. So glass electrode is what type of electrode? Glass electrode is an example of ion selective electrode. Can you define ion selective electrode? Ion selective electrodes are those electrodes which are selectively sensitive to a certain species of ions present in the solution and develops a potential against these ions, neglecting other ions that are present in the solution. Can you elaborate your answer? Yes, if I have taken a solution containing sodium ions, hydrogen ions, magnesium ions, and I will dip a glass electrode in this solution, the electrode will develop potential only against the hydrogen ions, neglecting the sodium and magnesium ions which are present in the solution. Glass electrode is made up of which type of glass? The glass electrode is actually made up of a long glass tube whose lower end is modified into a form of a bulb. Now this bulb is made up of a special kind of glass called soda glass, which has a low melting point and a high electrical conductivity. And the composition of soda glass is oxides of calcium, sodium and silicon. Where does the potential develops on the glass bulb? The potential develops at the interface. That is, at the membrane, between the membrane and the solutions on either side of the glass bulb. What is filled in the glass bulb? The glass bulb is filled with 0.1 normal hydrochloric acid. Tell me, what are the applications of pHmetery? pHmetery is very important and it should be determined for agriculture science, that is soil science, pisciculture, aquaculture, food processing industry, drug industry, municipal supply of water, and other industries also, such as paper, paints, coloring industry, cement industry. So almost all industries need determination of pH. We can also say the body care products also require determination of pH. Can you tell what are the advantages of pHmetery? pHmetery is very advantageous because it gives instant results. Even small quantities of solution can be used for determination of pH. We don't require large quantity of solution for its determination. The solution, even if it is turbid or colored, we will get accurate pH values. Can you tell me that how the pH you are going to determine with the help of this titration performing on the pHmeter? After completing the titration, that is addition of base from the burette to the acid solution, we will plot the curve by taking the pH values on the y-axis and volume of base on the x-axis. And then we will go for more accurate equivalence point by plotting a differential curve, that is delta pH by delta V on the y-axis and volume of base added from the burette on the x-axis. Once we have determined the pH, half of the equivalence point of pH on the y-axis will give us the pK of the given acid. The glass electrode you said you are using. So what is the internal reference electrode in the glass electrode? The internal reference electrode in the glass electrode is silver chloride electrode. So I think I have discussed almost all the viva voce questions that could be asked. If you have more questions, don't hesitate. Leave them in the comment box. Subscribe my channel. Keep watching my videos and don't forget to like and share them. It's very important to learn online. You can download the videos and keep watching at your leisure. Thank you very much. Take care of yourself.